Hello and welcome everyone to this podcast. In this podcast we're going to talk about protein control. So let's take a few steps back and let's draw a piece of DNA. And we're going to talk more specifically about DNA in a couple of sessions. But for now, remember that within the DNA there are genes. And these genes make a messenger RNA. And then this messenger RNA is made into a protein. I'm just going to draw the protein like this for now. We learned that this protein drawn here is the primary structure. We then learned that it could fold into a secondary structure like an alpha helix. We then learned that this alpha helix or the beta sheets could then kind of fold upon each other into a tertiary structure. And then we learned that these tertiary structures sometimes can form quaternary structures, where there's multiple subunits in one protein complex. We talked about how the tertiary or the quaternary structure could lead to a function, and we talked about protein function. How specifically enzymes can bind ligands, and upon binding those ligands they can catalyze reactions. In the immune system, they can target pathogens for destruction. So we spent a little bit of time talking about function. Now, we don't want this function, as great as it might be, we don't want it to occur in all cells at all time. This function has to be regulated. Now, we can regulate how this protein will function at many different levels. We can regulate it at the gene level. Does this gene have access to the enzymes necessary to move to the next step of transcription? So we can regulate here at transcription. I'm just going to put TX in. It could be regulated at the level of translation. It could be regulated here at the level of protein folding. Or once it's completely folded, we can regulate it at this level here of, I'm just going to put at the level of protein function. So once the protein's been made, can we turn it on and off? Primarily, that's where we're going to focus today. These areas up here, you'll learn more about in genetics, or if you've already had genetics, you've already learned about that. We're going to focus primarily here and a little bit here. And in doing so, there are 5 areas we want to focus on. The first is feedback inhibition. The second is allosteric regulation. One of my favorites. The third one, phosphorylation, a very important one. Our fourth one here, nucleotide binding, and really what I'm talking about here is GTP or ATP binding to proteins to establish some sort of function. And the last one here is something your book calls protein machines. I want to talk about these as protein complexes or factors. Some terms we often will interchange, complexes and factors. Okay, well, let's go ahead and get started and talk about feedback inhibition to begin to understand how we're going to regulate the activity of this protein once it's been formed. Right, feedback inhibition. In talking about feedback inhibition, let's draw out a simple pathway here, where you have some reactant here that we're just going to call A. And A is turned into product B, and then product B will be the reactants to generate product C. However, product B can also make product X. X can make Y, and Y can make Z. Each of these steps are controlled by an enzyme. So enzyme 1 makes the product B. We'll say enzyme 2 makes product C. Enzyme 3 makes product X. Enzyme 4 makes product Y. And as I'm sure you will be shocked to find out, enzyme 5 makes product Z. How does this pathway know to make X or C? How does it know there's enough X or there's enough C? Well, there's a lot of factors that might go into it, but what we want to talk about here is how the products of one pathway can influence that pathway or perhaps even another pathway. But for this made up pathway here, let's say there's a preference to go down this pathway to make Z. But once you have enough Z, it provides a negative feedback inhibition. And this negative feedback inhibition, by some mechanism, inhibits enzyme 3. With enzyme 3 being inhibited, now B can be converted to C. I'm going to say it again. The formation of Z provides a negative feedback inhibition on enzyme 3. This allows B to now be converted to C. Okay, this provides a nice example of negative feedback inhibition, where the product negatively impacts the pathway. So now let's talk about a positive feedback mechanism. And let's use this top part of our pathway. Let's stay in the same pathway, so to speak, here to describe it. And let's just say that product C here, as it builds up, comes back here and somehow enhances the activity of this enzyme. So this would be a positive feedback. The difference between negative and positive is that negative inhibits an earlier step in the pathway, whereas positive enhances an earlier step in the pathway. So as I've drawn this here, it's a fairly straightforward mechanism involving 5 different enzymes, 6 different reactants or products. However, I'd like to stress to you that if this were a real biochemical pathway in a real cell, it wouldn't be quite this simple, because there would be other pathways that might feed into A, or might feed into C, or might inhibit Y, or might take Z to a different pathway to regulate that pathway. So as we talk about these terms throughout the semester, remember that everything's connected, that it's really hard to manipulate these pathways without an appreciation for everything that's happening inside of the cell. You really need to understand the biochemistry of what's happening in the cell. Alright, so as we look at this, there might have been a couple questions that popped in your mind. The first question that might have popped in your mind as we were discussing this is, why does B, this B here, prefer enzyme 3 over enzyme 2? You have the same substrate, but it appears to prefer to bind to enzyme 3 until it gets inhibited. But the only way it's going to bind preferentially to E2 is if we somehow inhibit E3. So why is that? The second question really is the focus of today. That is, how does Z, this Z here, inhibit or regulate, might be another way of saying that, E3? Or how does C, this C here, enhance the activity of enzyme 1, this enzyme? And we'll address this question in one way or another as we talk about the other things that are involved in controlling proteins. Okay, let's begin this next section on allosteric regulation. I like to look at the breakdown of words sometimes to give an idea of what we're talking about. So allo means other, and steric here, or this the S-T-E-R, means shape. So when we're talking about allosteric regulation, we're talking about regulation that changes the shape of the protein. And it makes good sense to talk about allosteric regulation right now, because we just talked about this pathway, and I'm going to draw a version of it here, or at least most of it. And we said that Z somehow inhibited the enzyme between B and X. So how could Z inhibit E3? Well, we can imagine several different ways, but I want to focus on an allosteric regulation. So let's draw enzyme E3 here in our classic enzyme model here. So this is E3. And let's say that B binds here. Up to this point, most scientists believe that inhibition of an enzyme was caused by some sort of competition. That is, Z would have bound here and outcompeted B for that site. But when they came across certain inhibitors, like Z in this case, that couldn't bind to this active site, And through biochemical studies, they could see it wasn't competing with that site, but it was indeed inhibiting it. They reasoned that there must be another binding site on this protein, where in our example here, Z could bind to this enzyme. Binding to this enzyme changed the way it folded in such a way that the original substrate, in this case B, could not bind here. So this is the classic example of a non-competitive inhibitor that we talked about last week. But now I'm bringing it to you in the context of protein regulation. So this product here, as made Z, comes back here, binds to an alternative binding site, changes the shape, makes a conformational change in such a way that the actual active site is no longer accessible to the substrate, in this case B. Now, this kind of regulation, this allosteric regulation, usually results in this negative feedback inhibition. You could, though, imagine that a product, if you remember we said C, somehow comes back here and enhances this enzyme. You could see, in a positive way, that an alternative substrate binds to this alternative site and somehow enhances the binding. Maybe it makes it bind with greater affinity. Maybe it makes it so it turns over faster. However it does that, you could see how changing the shape could also have a positive effect. So to recap here, allosteric regulation is when a molecule of some kind, one of these products here, can bind to an alternative site. It changes the shape of the protein. As it changes the shape of the protein, it can either prevent an enzyme from doing its job, or it could enhance the enzyme in doing its job. Now let's move on to the next topic of protein regulation. And this next topic is phosphorylation. I want to keep this pathway written right here, because maybe we can refer to it again. To begin our discussion of this, I want to point out how important phosphorylation is. In any given cell, at any given time, there are, give or take, about 10,000 proteins. When we look at these 10,000 proteins, greater than 3,000 at any given time are phosphorylated. Over a third of our proteins in any one of our cells undergoes phosphorylation, indicating that this step here of adding a phosphate group to a protein is probably pretty important. So let's talk about this in greater detail then. And this is a convenient one to talk about right after allosteric changes, because sometimes the process of adding a phosphate group will change the shape of the protein. So this is an example of how we can change a protein's shape. So what is phosphorylation? Well, let's say you had a protein here, and I'll just draw a folded protein of the amino acids on it. Sometimes we can add a phosphate group. Many different amino acids can take a phosphate group, but two big ones that we know a lot about are serine and tyrosines. Throughout the semester, we'll focus on different aspects of serine and tyrosines being phosphorylated and the importance of those phosphorylation events. So we're going to use those as examples here. But there are examples of threonines, histidines, arginines, lysines, and other amino acids also being phosphorylated. Alright, so let's take our serine here. And to remind you, serine has an R group that looks like this, CH2 and then a hydroxyl group. Serines can be phosphorylated, and they're phosphorylated using an enzyme called protein kinase. And when they're phosphorylated, they look like this. With a phosphate group added to the oxygen of the hydroxyl group. The most notable thing that this does here is it adds two negative charges. It makes it much more negative. Another way you could look at this is that by adding this phosphate group here, these two negative charges, it makes this already polar amino acid more polar. Now this is a reversible reaction, and the enzyme that removes this phosphate group to recreate the hydroxyl group is called protein phosphatase. Protein phosphatases remove phosphate groups. Protein kinases add phosphate groups. Typically, adding a phosphate group makes the protein active. And so that's how I always remember that protein kinases add the phosphate groups to make them active. Because I think of kinases and I think of kinetic energy. Kinetic energy is the energy of action. And so I just, in my weird way of thinking, that's how I've always been able to remember that kinases add phosphates, which create the active form of the protein. Okay, I want to come back to these two negative charges here and ask the question of how could increasing the negative charge change the protein? Well, by adding these two negative charges, as I said before, you make it more polar, but now the protein can interact with positive charges on other molecules or itself. So for instance, it could interact now more easily with lysines or arginines. This could change the way the protein folds. So let's think about this as to how this could impact this pathway here. Let's say that enzyme 3 here is only active when it's phosphorylated. Upon being phosphorylated, it folds in such a way that it can convert B to X. And let's say the way we inhibit it is that Z here acts as a phosphatase or activates a phosphatase somehow. It comes along here and removes this phosphate group. It now folds back to its inactive form and can no longer convert B to X. Alternatively, maybe enzyme 1 here needs to be phosphorylated to be fully active. Maybe it has some activity without being phosphorylated, but maybe being fully phosphorylated enhances its activity because it folds in such a way to make that active site more accessible to B. And in this scenario, maybe C acts as a kinase, or maybe C activates a kinase. But the end result is that C, by its production, adds a phosphate group to E1, creating this one here, and enhances this activity. Again, adding a phosphate group, removing phosphate groups, can allow it to interact with these positive molecules and can change the shape of these enzymes to make them more active or less active. Another way these positive charges can help is through something I'm going to call scaffolding. So in talking about scaffolds, I want to draw something over here. Let's say we had two monomer proteins that were non-functional on their own. But let's say adding a phosphate group to one of them, adding that negative charge, enhances the ability of these two to bind. And so now they're connected here at this phosphate group. And now this dimer that I just drew here is functional. Okay, the next thing I want to talk about is nucleotide binding. And with this I mean ATP or GTP. Proteins can bind ATP or GTP, and in doing so influence the activity of that protein or enzyme. I want to start with GTP though. Adding a GTP molecule to a protein is similar to what happens with phosphorylation, but there are some differences. For instance, like phosphorylation, it adds a phosphate in the form of GTP, guanine triphosphate. However, unlike phosphorylation, when we add a GTP, it doesn't covalently add it. So this is more of a non-covalent interaction, where adding a phosphate group is a covalent interaction. Adding a GTP influences protein or enzyme activity or function, I could say. Lastly, just like with phosphorylation, it also causes an allosteric change. In many ways, this acts as, thinking about number 2 here, it acts as a molecular switch. So you can think of a protein here that has GTP bound to it as being active. If we're thinking about a switch, we would think of that as being on. Now this GTP can be hydrolyzed, in which case the protein here will be bound to GDP, guanine diphosphate. This will make it inactive or off as a switch. Then what happens is the GDP is released and you just have an empty protein. This would also still be inactive or off. But we can turn it back on. We can switch this protein's activity to the on switch again. And we do that by having it bind GTP again. It's active on. And we'll talk a lot about this when we talk about signaling pathways and endocytosis and some other pathways. I want to mention one more thing here about how GTP turns this enzyme activity on and how it turns it off. Maybe in the on state, the conformation is something like this. And then when that GTP is hydrolyzed, this sort of door I drew here shuts, changes the conformation in such a way that access to the protein or enzyme has been affected. So again here, the door I drew is off, is shut. And then to turn it on again, the GTP binds and opens the door, once again activating it. The key is that GTP changes the conformation of this protein. When GTP is bound, it has conformation that is active. When it's not bound, it's a conformation that is inactive. Okay, I'm going to stay with the nucleotide binding, but I now want to talk about ATP, adenosine triphosphate. The ATP that we've learned about in many classes that we often call the cellular energy. A few moments ago when I talked about phosphorylation and binding of GTP, I talked about how that could change the conformation of a protein and how that could lead to differences in enzyme activity. ATP also changes the conformation of a protein, but I want to talk about it in its role in movement. We can think about how ATP is involved in muscle contraction, in vesicle or organelle movement in the cell. And ATP is very important for this. So how does it do this movement? It moves vesicles and fibers for muscle contraction, and I'm just going to focus on how ATP can be used to move things within the cell. So how does ATP help move vesicles or organelles within a cell? It does so using something called a motor protein. Motor proteins bind ATP. ATP is hydrolyzed, and that will change the conformation of this motor protein. And just to draw it generically, I'll go from a circle to a square. What I want to do right now is to show a quick video, and in this video, which we've seen before, I want to focus in on one key area that will show how hydrolyzing ATP, or rather it will show how changing the conformation of the motor protein allows it to move. So let's watch that and I'll come back to the whiteboard.
Okay, here's the video I wanted to show you. You've seen it before. I showed it at the beginning of a class earlier in the semester. But as I hit play, I want you to watch this structure here, and this is the motor protein, and this is the vesicle. And I want you to watch how it's moving along this microtubule here at the bottom, carrying the vesicle with it. And I'll say a little bit about it as it's moving. See how it's moving forward each one. It looks like it's taking a step. Just moving forward. And what's happening here with this protein here, this motor protein, is there are conformational changes in these structures here that look like legs that are moving the motor protein down this cytoskeleton carrying the vesicle. I'm going to back up a little bit. And what's happening here, it's binding ATP, hydrolyzing ATP, and then binding ATP again. Every time it binds an ATP molecule and hydrolyzes it, it changes its conformation. And as it changes its conformation, it moves down this microtubule. Okay, I'm going to now go back to the whiteboard and we're going to talk a little bit more about...
Okay, welcome back. Now, I think that movie is a good example of showing how that vesicle can move across the cytoskeleton and how it requires a protein conformation change. But it doesn't really show when or where ATP is binding. And so I'm going to try to draw this here. We'll see how it works. But it really does so in a manner to what you saw on the video. It really does look like it's walking. So these motor proteins, let's draw them like so, carry this vesicle on top. In order for it to get down here, it's got to move along the cytoskeleton. ATP will bind to this now, and when it binds to these motor proteins, what happens is it changes the conformation. And we'll do a little time-lapse photography here. So it binds the ATP, and this part of the motor protein now comes down and touches the cytoskeleton. I'll draw the vesicle on here again, but I want to make sure I draw ATP here, bound to the motor protein. Okay, so now the ATP is hydrolyzed, and what it does, as it hydrolyzes it, this part of the motor protein kicks up. So it moves up, kind of like stepping up. This part of the protein has a conformational change and lets go of the cytoskeleton, and then repositions downstream on the cytoskeleton, like so. Still carrying the vesicle. And at this point, ADP and the phosphate that was just hydrolyzed is still attached here. And I'm out of room here, sadly, so I'm going to draw the next stage just on the same cytoskeleton. The ADP into the inorganic phosphate now is released. Upon releasing, what happens is this part straightens up, moving the vesicle forward, like so, and in doing so, this one here kicks up, so to speak, carrying the vesicle with it. And remember, now the ADP has been released. And it will continue to do this. Now this will bind ATP, this will step down, it will hydrolyze, and this will move forward. And it'll continue this cycle until it gets to its final target. And we'll talk more about this when we move into our discussions of the cytoskeleton. But again, as with everything else we've talked about, the key to this is, upon binding ATP and hydrolyzing ATP, there is a protein conformational change. And in this case, it enhances movement. Whereas when we talked about GTP and phosphorylation, we gave examples of enzyme activity and protein structure. The last thing I want to talk about are protein machines. And in essence, we've touched on this earlier, and that is how proteins function when they form a quaternary structure. And if you remember, I talked about how one monomer and another monomer come together to form a dimer, and it was this dimer that was active. But I want to take it up one more level, and I want to bring in a term that sometimes we throw around, and it's called a factor. And specifically, let's talk about a transcription factor. Those of you who've taken genetics, you've heard of these. Those of you who will take genetics will hear of them. Those of you who don't plan to take genetics should plan to take genetics. So you can learn more about these awesome things called transcription factors. But what I'll tell you they do now is that they take a gene found on DNA, and these transcription factors will help find that gene. When they find that gene, and I'm really simplifying this because I don't want to spoil too much of it for when you take genetics. So these transcription factors then will release a messenger RNA that is then made into a protein through translation. Now, you don't need to know any of this for the test. I'm just trying to introduce to you what a transcription factor does. It binds to DNA, it recruits a lot of enzymes and other proteins here to make this RNA. But I want to focus on this term factor. This is really what we mean when we say a protein machine or a complex of proteins. So for instance, this transcription factor is not a single protein. It's many proteins. Some factors can have as many as 20 proteins, some with more in them. And I'm not going to draw all the transcription factors found in this particular transcription factor, but I want to illustrate how these many proteins come together and work as a single unit. So maybe we have a central protein here, and maybe we'll call it an ATPase, something that hydrolyzes ATP to generate energy. Then maybe there's another protein here, and this protein is a DNA binding protein. And then let's draw another subunit, and maybe this one binds another transcription factor. Okay, so this is a transcription factor, but maybe in this process we need to get a lot of transcription factors here, which is true, we do. So this is another transcription factor. Maybe this one here binds RNA polymerase, which is ultimately the enzyme that makes the RNA. And maybe this one here, I'm going to call X for now. I'm going to come back to that in a moment. But let's say it provides another specific function for this protein, or rather for this factor, I should say. Together, and again I simplified this, there are more proteins that bind, but together this makes up this specific transcription factor that binds the DNA. So the question might be is, why? Why do it this way? Why not just have these 5 proteins, which really would be 20 if I drew everything here? Why have these 20 proteins just separate and they can bind here independently? So let's just write why here. One of the reasons is it eases the regulation. You don't have to regulate 20 different proteins and their ability to get here. You have to regulate one of these proteins, maybe it's a DNA binding protein, to find here. And once they find this spot here, they bring all these other proteins with them. And once it's there, now these others take over. So it kind of creates this kind of a division of labor. Many proteins are here. They all have their own function. They all do their own job. Okay, now there's a third reason why we want the ability to form these big complexes. And in talking about it, let's mention a couple things here. Within organisms like animals and plants, we have many different tissues. And so let's just take an example where you have, say, a liver tissue and you have a pancreatic tissue. And in the liver, we want to make sure we activate liver genes. And in the pancreas, we want to make sure we activate pancreas genes. You don't want pancreas genes to be turned on in the liver, and you don't want liver genes to be turned on in the pancreas. And so now I want to come back to this mysterious X subunit I put here. It's possible, in fact it's probable, that all of these proteins in this complex, this factor here, are necessary to turn on and activate genes in any cell in your body. But we don't want to turn on every gene in the body. We only want to turn on specific genes. We only want to turn on liver genes in the liver, pancreatic genes in the pancreas. This subunit could perhaps provide that specificity. So in the liver, we would have the liver specificity protein added to this complex, so that it would turn on liver genes. It would bypass any genes here that are pancreatic genes, that are brain genes, that are heart genes, but it's going to have the specificity to know to turn on specific genes in the liver. Likewise, in the pancreas, you're going to have this exact same complex, except you're going to have a subunit that specifically targets pancreatic genes. I have simplified this quite a bit. It's a little bit more than just this one subunit that comes in and comes off. There's a much higher degree of regulation. But the point I was trying to make here is that by having these multimeric complexes here, you can exchange certain proteins from it, retain all the other protein function, but add a new function to it, which could be tissue specificity. It could be environmental changes. It could be a developmental process where you want to turn certain genes on in a embryo that you don't want to turn on as an adult. It allows to use the same protein complex but in different situations. So I'm going to write that down because I think that's key. Allows to use same complex in different situations. Or I probably should say similar complexes, because we did change one protein here. Okay, that's all I have for this podcast, but I'd like to close with a quick summary. In this podcast, we talked about 5 different ways that proteins can be regulated once that protein has been made. First, we talked about negative and positive feedback control, how a product can either inhibit or enhance an earlier step. We then talked about how these feedback mechanisms often exert their change in activity by an allosteric change, and how that allosteric change is essentially just a conformational change of that protein. We then talked about phosphorylation as an example of how adding a molecule to a protein, like a phosphate group in this case, can cause an allosteric change that affects the activity of a given enzyme or protein. We also talked about how ATP or GTP, 2 nucleotides, could be added to an enzyme or to a protein to change its conformation and to lead to changes in activity, protein structure, or movement of things within the cell. And then we closed with these protein factories, how having multiple proteins coming together into a single factor has many advantages to a cell. Okay, that's all I have for this podcast. So if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.